Hello, my name is Osval Baral, and I'm a postdoctoral fellow at the University of British Columbia. Today, together with my colleagues Autry Chatterjee and Tom Sorosky, we will be presenting our video pitch on speech and eye movements for early detection of Alzheimer's disease.
Hi, dementia is a progressive neurodegenerative process resulting in impaired cognition, loss of functional independence, and eventual death. It affects 47 million people worldwide, and it is one of the costliest diseases in the developed countries. Over 747,000 Canadians live with Alzheimer's disease or another form of dementia. Despite the prevalence and impact of Alzheimer's disease, there are still no treatments that can stop progression. Current evidence shows that the brain changes characteristic of Alzheimer's disease, including neurofibrillary tangles and amyloid plaques, are present years before overt symptoms appear. A digital biomarker to assess disease risk and progression will not only help identify early-stage individuals, but could also reduce the need for more invasive procedures like lumbar puncture and PET scanning in the context of early disease staging. Changes in acoustic and linguistic speech features have been identified in individuals with Alzheimer's disease and mild cognitive impairment. Changes in language include semantic and lexical content as well as syntactic complexity, which decline with disease progression. Eye movement impairments are also present from the early stages of Alzheimer's disease. People with mild cognitive impairment demonstrate abnormalities in prosaccades, fixation, and refixation as compared to healthy controls. People with Alzheimer's disease also display disruptions in multiple types of basic eye movements.
Our primary research question is, can we identify early-stage Alzheimer's disease from speech and gaze data using machine learning? Today, to answer this research question, our research team has recruited and administered an assessment to collect simultaneous speech and gaze data of 73 healthy volunteers from the community and 68 patients from a memory clinic with either mild cognitive impairment or Alzheimer's disease. During the assessment, participants are asked to describe the cookie theft picture from the Boston aphasia battery, while their gaze and speech are recorded using an infrared eye tracker and an audio recorder. In addition, participants are also asked to read out loud a paragraph and recall a positive memory, conforming a total of 3 tasks in the assessment. We then perform feature engineering on the signals to extract information that could be discriminative between patients and controls in our dataset. Next, we feed the speech and eye features into machine learning algorithms to train classification models that will allow us to predict, given the speech and gaze data of a participant, whether they are healthy controls or have some form of cognitive impairment. Results achieved so far are very promising, and the best performing models achieved 0.8 area under the curve. In fact, best classification performance was achieved when combining speech and gaze signals, outperforming baseline models that use speech data alone. We recently published these findings at the Machine Learning for Healthcare conference. The work so far involved the analysis of the cookie theft picture description task, and with support from the CCNA, we will be able to apply novel machine learning techniques to the rest of the tasks in our dataset. The goal is to evaluate whether adding several complementary tasks in our machine learning models leads to improved classification performance. Another critical step is to extend our dataset longitudinally by repeating the testing battery over 6 months over 2 years. We have started this effort already, and with the support from the CCNA, we can ensure that this work is further carried out. The goal here is to gain understanding into the role of speech and gaze in early Alzheimer's disease and how they may indicate disease progression over time. Another key step will be increasing the size of our dataset. For our preliminary analysis, we grouped individuals with mild cognitive impairment and Alzheimer's disease into a single patient category. Funding from the CCNA will support targeted recruitment, allowing us to increase the number of individuals in our cohort with mild cognitive impairment. This in turn will allow to train more sensitive machine learning algorithms that can distinguish individuals with mild cognitive impairment from those with Alzheimer's disease. To disseminate our biomarker to researchers and clinicians, we need to determine its tolerability and feasibility in our target population. To date, we have administered a questionnaire to 56 patients and 56 controls, seeking feedback about various aspects of the assessment. The vast majority of participants were comfortable, relaxed, and willing to repeat the assessment on a yearly basis. very few participants had privacy concerns with the technology, as we ensured that participants were aware of their data being recorded and how it was being processed. Developing this questionnaire with input from the CCNA Knowledge Translation and Exchange team will help us further demonstrate tolerability. Furthermore, input from the KTE team will help accelerate the dissemination of our findings to people living with dementia and their families, as well as clinicians and other researchers. We will present our findings at the next UBC Alzheimer's Public Forum, an event designed to update patients, families, and the public on the current neurodegeneration research. KTE input will help guide the creation of a video aimed at study participants, explaining our research in lay language, encouraging study participation, and sharing our preliminary findings.
With the help of the CCNA's Ethical, Legal, and Social Implications team, we aim to shed light into several important questions related to the use of a machine learning model in clinical and research settings. For example, machine learning models are complex and constantly changing with new data. How does a changing model impact its use as a biomarker? Furthermore, the generalizability of any machine learning model depends on its training data. An ideal machine learning model can be used across many populations, producing similar results. We aim to understand our model's generalizability by comparing its output in a number of different populations. As well, biomarkers typically correspond to a particular disease process. However, as our models are generated using a number of complex mathematical transformations, some highly predictive features may not be clearly associated with the disease process. This could mean that highly predictive features may not be interpretable. In addition, recording user video, voice, and eye movements, and subsequently using speech-to-text software could raise ethical questions about privacy. Following the speech gaze assessment, we asked participants if they had any privacy concerns using the technology. 5% of memory clinic patients and 2% of healthy volunteers stated that they had concerns. With ongoing speech gaze assessment, we'll continue to assess participant privacy concerns with the technology. How does our work align with CCNA strategic priorities? A digital biomarker that identifies early stage neurodegeneration directly addresses the CCNA's prevention priority. Our digital biomarker will identify individuals who are at high risk of developing dementia. In addition, it will also identify gaze and speech features that are most discriminative for detecting early stage dementia. Identifying at-risk individuals allows for preventative treatments and lifestyle modifications to start earlier, when they would be likeliest to have an effect. Early identification also allows for longer engagement and shared decision-making regarding life planning, healthcare, and involvement in research. We are an interdisciplinary team of clinicians and computer scientists with expertise ranging from neurology, clinical trials, and occupational therapy to natural language processing, user modeling, and artificial intelligence. Our team is composed of CCNA investigators, Dr. Thalia Field from Team 7 and Dr. Robin Shung from Team 8. These investigators are responsible for supervising our team's trainees, Dr. Otri Chatterjee, Dr. Oswald Baral, and myself, Tom Sarosky.
Our clinical team selects and screens eligible participants for the study, administers the speech and gaze battery, and collects questionnaire data. Our computer science team is in charge of cleaning and analyzing the data, implementing appropriate AI models, as well as evaluating the performance, while ensuring that the output has a large degree of explainability, so that in turn, the medical team can make sense of the output and derive clinically relevant insights.
Funding from the CCNA will help support ongoing training and capacity building within our existing interdisciplinary team. To date, our trainees have had the opportunity to disseminate the team's work at national and international interdisciplinary conferences, which is a critical aspect in the development of their academic career. CCNA funding will allow our trainees to continue this active participation in academia by contributing to future manuscripts and knowledge exchange activities, such as workshops and conferences.
So in conclusion, our team is using machine learning to develop a novel digital biomarker which will assess risk and progression of Alzheimer's disease. The goal is to design a simple, accurate, and non-invasive screening tool which will help identify early-stage individuals for clinical trials and reduce the need for more invasive procedures. Support from the CCNA will enable our trainees to generate further insights from our dataset and will ensure ongoing data collection, leading to improved training and capacity building of our trainees and active participation in knowledge translation activities. Thank you very much.